just to start, let's see, the Levin in the title, of course, is Don Levin, and the 35 years refers to the fact that it's 35 years ago since he published this absolutely seminal paper. Hello? Okay. Which has been widely cited and certainly was really instrumental in my development as a scientist. In that paper, he said his purpose was to show that autopolyploidy greatly alters cytology, biochemistry, etc., all sorts of things, and that autopolyploidy was actually a major force in evolution. And by autopolyploidy, he meant genome doubling without genome merger. He specifically contrasted with hybridization, heterosis, and things like that. Okay, so he also said the theme of a positive nucleotypic effect will be developed. How many people here know what the nucleotype is? Okay, not that many. And so the nucleotype is a term coined by Mike Bennett back in 1971. It describes those conditions of the nucleus that affect the phenotype independently of the genotype, essentially. So, nucleotypic effects include things like cell cycle and things like cell size. In particular, cell size is very important because cell size is central to most concepts of what polyploidy genome doubling actually does. And so this goes back to Munsing, for example, who actually cited in earlier work to talk about the fact that cell size has a strong influence on morphology and physiology. And it's really where things start with polyploidy in most models of what autopolyploidy does. And here's a more recent paper, very nice review paper, showing the centrality of increased cell size with morphology, physiology, and everything else downstream. But the question is, what causes polyploid cells to be larger? Is it polyploidy? Is it the genome size? What is it exactly that's doing that? Now, just a quick digression, which I don't have much time for, but I'll just say that I certainly thought as a systematist that I could just go to the literature on cell biology and find this out. Okay? It turns out that that's not so easy. So just to say again, in recent reviews, the mechanisms by which polyploidy might actually do something remain a major challenge. And so Jeremy and I have been working for 2 years to try to update just the first part of Levin's paper, where he talks about the influence of polyploidy in cytology. So this is something that's going to be coming out, I hope, in early 2019. You can look for it there. And I call it polyploidy cell biology, not so much for dummies, but by dummies. Because it turns out we had a whole lot to learn. So that's what I'm going to share with you today. We cover a wide range of topics in this very large review with more than 500 references, and it's been a lot of work. It's either the most stupid thing I've done in my career or the best. I'm not sure which. Today I'll give you the Cliff's Notes version, essentially. Okay, so starting at the top, what I'm not going to talk much about is just the obvious things about the transcriptome and proteome, how that's affected by gene dosage changes. But that's clearly a very important part. We know that the genome is downsized, at least over time in polyploids. Jeremy and I have been interested for some years about what happens to the transcriptome, how it also is downsized and much more rapidly than the genome. The proteome remains something that we really don't know a lot about yet, so in terms of quantity. So here's the beginning of things: polyploidy, increased gene dosage, increased bulk amount of DNA. The dosage here leads to larger transcriptomes, but it's not an obvious effect, and there is some downsizing going on. That relationship with the proteome presumably is related somehow to larger transcriptome as well, although not really well understood. And today I'm mostly going to talk about this part here, the increased bulk DNA content, the nucleotypic effects. So this turns out to be rather tricky because size itself is something that's not obvious when you think about measurement, whether it's volume or mass or area. These are all things that get called size, and so when you say a cell gets larger, it's larger size, what does that actually mean? That turns out to be rather difficult. What determines cell size? This is a 2012 review with an absolutely all-star cast ranging from people working on yeast, plants, mice, Drosophila, C. elegans. I think everything is in there. They've included, we still don't know how cell size is determined. And Robert Doyle is here, he's been around a long time. And so cells have been around for some time. Now this is a 2017 review, so 5 years, a lot has happened in cell biology. And, but we still don't really know what controls cell size, and this is important for saying that. What about the nucleus? That might be an obvious sort of thing, right? Here's Robert Brown, 1831, discovered the nucleus. If you want to know the state of things, just ask the question. So basically, we have a sort of problem here. very simple model would be more DNA equals more chromatin and leads to a larger nucleus to accommodate. That seems like a very obvious thing, but the conclusion in very recent reviews is that ploidy changes may contribute to nuclear size determination, but it only is minimally so. So it's really not such an obvious thing. This is a too simple model, it turns out. Okay, and our understanding of how nuclear shape and size are regulated is rather poor. Another recent review in plants, shape of the nucleus changes quite a bit across different cell types, size changes. All of this is not understood at all. All right, so if cells and nuclei get larger, how might that affect things? All right, that's the next question. Well, the classic answer is surface to volume ratio. Because the area increases as the square of the radius, the volume increases by the cube of the radius, and so if you take a diploid cell and then double the volume of the cell, the surface area does not increase by as much. And so Levin and before him others, and since him many people as well, have said that there are manifold repercussions at the cellular level for this reduction in rates and all sorts of other things that can happen. So this is well known. Now, it turns out that's not so easy. Jimmy Walba, in that sort of all-star paper, also pointed out that this doesn't take into account the plant vacuole and the fact that it's really vacuole with area, and those surfaces are also very critical in thinking about how surface-to-volume ratios may change. Okay. And here's another one, cell size control, a mechanism for maintaining fitness and function, a 2017 review. Distance is very important. So here, size-induced increases in intracellular distances could limit almost all cellular functions. The rates of biochemical reaction would be reduced, and metabolic changes would occur. But unfortunately, in 2017, there's little quantitative evidence regarding the extent to which increased intracellular distances affect growth rate, metabolic rate, or cell size. So again, it seems obvious that distance should be important, but we really don't know how. All right, so one of the things where this is going to happen is in the nucleus after transcription, here and along the sides here, mRNAs are assembled into nestor-ribonuclear proteins, which diffuse through the interchromatin channels to the core complexes where they're actively transported. So how will size change that in volume? So first of all, surface to volume effects, nuclear pore density relative to volume. This is actually something that Levin mentioned in 1983. There are very, very few measurements about this at all. And the second is diffusion distances will change. But it's not even clear how diffusion works in the nucleus. This is another area that's just coming under scrutiny. We also know that you can change the density of chromatin, and this will have important consequences as well. There's a recent paper looking at the size of diploid and autopolyploid nuclei in Arabidopsis and concluding that the density of chromatin increases with DNA contact. That has important consequences for folding, and folding is regulation, as it says here, so you can have effects on the transcriptome as well. It's interesting that nuclear architecture varies across species and across cell type within species. So you really can't talk about one particular effect. You have to think cell type by cell type and species by species, and that's maybe different in different organisms. The chromatin positioning, compaction, and so on is dynamic, and so you can see that there are going to be effects if you change any of these parameters. We're finding out that light and abiotic stresses regulate or change the transcriptional state of plants' cells by actually changing the dynamic nature of chromatin and changing the positioning of chromatin. So here's some recent papers on that as well. Polyploids often show greater tolerance to abiotic stresses. Is this part of the answer to that question? Cell organelles are all interconnected. I didn't know this. I don't know how many people know this, but they're all interconnected all across everything. And size and number of organelles changes with polyploidy. So you're getting all kinds of other effects possibly through that as well. And the cytoplasm itself is highly crowded and highly structured. This is what the cytoplasm looks like. The cell is not a watery bag in which enzymes flow freely. There are questions even about whether diffusion works the way that most people assume it does for even experimental purposes. So looking at cell size is connected to protein concentration, cell growth, gene expression. All these things are interconnected, and changing any of them can change the others, and polyploidy probably changes all of them in some way or another. We don't really know how. So here we show this. Here's our question mark, larger cells, larger nucleus, more chromatin. All these things are strongly correlated. But then you think about, well, the larger transcriptome may play into these things, and the proteome certainly feeds into larger cells. We have longer cell cycles leading to larger cells, potentially, and larger cells needing a longer cell cycle, which direction do the arrows actually go? We have increased dosages that affect the cell cycle as well by specific genes. We have metabolic changes that feed back in various different ways. We have larger or more organelles which also change metabolism. Mitochondria are intimately associated with this, and so is the nucleolus and ribosome numbers. So I'm going to end with about 17 key questions. I'm not doing the time, but anyway, here we go. Just stop me when I've run out of time here. So how do cell size and nuclear size, whenever you measure it, and karyoplasmic ratio, that's the nuclear to cell size ratio, vary across cell type and species? How are these affected by polyploidy? How do duplicate chromosomes and autopolyploidy associate with the nucleus? Are they grouped together, partitioned separately, and how does that all work? Because again, the position of chromosomes in the nucleus is really important for regulation. How does genome doubling affect intrachromosomal associations? We're finding out that there are transcriptionally associated domains in plants now. This is important too in terms of what polyploidy does. How do the various transcriptomes, and there are lots of different transcriptomes to consider, by the way, not just the transcriptome as we usually think of it, vary with cell type. How is it affected by ploidy? How does size change of the transcriptome in different species for a given cell type? Does an increase in transcriptome size drive increases in nuclear or cell volume, or vice versa? How does nuclear crowdedness vary with nuclear size across cell types and species? How is that affected by polyploidy? Does the number or density of nuclear pores change? For a given messenger ribonucleoprotein, how does the time from transcription to transit through the nuclear pore complex vary with cell type, species, and ploidy? How does the rate of import into the nucleus, transcription factors, ribosomal protein signaling molecules change with ploidy and size changes? For a given mRNA, what's the average time of translation once it gets out of the nuclear pore complex in diploids versus polyploids? Is mRNA stability affected by polyploidy? What effect, if any, does polyploidy have on translational regulation of gene expression? Is protein stability affected by polyploidy? How do protein, metabolite, and ion abundances and concentrations vary with cell type, and how are these affected by polyploidy? Is cell type specific size response to polyploidy conserved across species? Remember, the architectures are different. How does cytoplasmic crowdedness vary with cell type across species, and how is this affected by polyploidy? How are the rates of intracellular and extracellular movement of DNAs, RNAs, proteins, and metabolites affected when larger cell sizes accompany polyploidy, and are any such changes biologically meaningful? For a given cell type, how does size and number of each non-nuclear organelle, lots of them are listed, correlate with cell size, and how does this relationship change with increased ploidy, and what biologically relevant effects might be predicted and tested from any ploidy-related differences? And how does polyploidy affect cell number, rates of endopolyploidy, and other parameters in addition to cell size that contribute to organ size and shape? The phenomenon of compensation, for example, how does that work in polyploids? So these are all questions. There are answers to, as far as I can tell, none of them at this point. And so I wish I were younger. Because I'm thinking about a second career as a cell biologist. But because of this, because of what's happening in the single cell data. You have 2 minutes? Oh gosh, this is much for me. Anyway, so yeah, there's so much going on right now. I just got some, the first from a colleague, some single cell transcriptomic data the other day, and the stuff you can do now is just phenomenal. So here are the technologies, and we're ready to go, basically, with this. Enjoy.